So without further ado, I'd like to invite Morgan Youngblood to talk about wastewater infrastructure.
Hello, thank you for coming. My name is Morgan Youngblood. I'm going to be presenting my research, investigating environmental health and sustainable wastewater infrastructure in Kailua-Kona, Hawaii, USA. The state of Hawaii has struggled to find a sustainable pathway for wastewater infrastructure, given the abundance of cesspools installed since statehood in 1959. This project takes place in Kona, where there are both an abundance of cesspools and a wastewater treatment plant with a sewage line that runs along the shore. My project is a part of a broader collaborative project with the Kahului Bay Education Center, the County of Hawaii, the University of Hawaii in Hilo, the Department of Land and Natural Resources, and Vai'vai Ola Waterkeepers Hawaiian. And the goal of the assessment is to pinpoint high-risk areas that are in need of wastewater infrastructure upgrades. My independent research evaluates Enterococcus facialis, a fecal indicator bacteria. It also looks for, evaluates correlations between fresh water sources and Enterococcus concentrations, turbidity, and cesspool density in the surrounding areas. So these are the 12 water quality testing stations that were selected. And then you have the cesspools are marked by the little dots there inland. The sites were selected based on lowest salinity levels along the shoreline. And so we tested samples. We took samples monthly at each one of the stations. We also took the temperature, dissolved oxygen, and salinity levels at each one of the stations as we were collecting water samples. We got triplicate samples early in the morning before they were exposed to UV light, and also when tide conditions were lowest. Each one of the samples I cultured for Enterococcus using IDEX testing methodology. In the lab, I also took the pH and the turbidity of each water sample. And then here are the results of, at the bottom here on the x-axis, you can see each one of the stations, starting with the furthermost north, the King Kamehameha Hotel, and then to the south end, you have the Keauhou Canoe Ramp. You see a few of the stations exceed the state standard of 130, most probable number per 100 milliliters. And then a couple of the stations exceed the 35 most probable number per 100 milliliter state standard. And the results of my Kruskal-Wallis ANOVA test are up there in the upper left. And then this is another way to view the same data as before, so you can kind of see these locations as hotspots of Enterococcus concentrations. These are average Enterococcus concentrations throughout the year-long study. You see a hotspot down there at Keauhou. Kahului is just north of that, if you're moving from the south up, a little cluster there. And then the orangish reddish one up towards the top is Numalu Hulahi'i Beach, and then the reddish one towards the top is a spot behind the Royal Kona Resort. And then I compared these to the cesspool density in each one of the areas, and I didn't find a correlation between Enterococcus concentrations and cesspool density in this study. I also evaluated salinity in this study for a correlation between Enterococcus concentrations. And then the gradient that you can see there is temperature, and we don't see a correlation. The results of the Spearman rank correlation test is up there in the top left, and we did not see a correlation between Enterococcus concentrations and salinity in this study. And then I do see a correlation between turbidity and Enterococcus concentration. This is likely due to high wave action and sediment that gets stirred up from that. And then I also wanted to note that we see elevated levels of Enterococcus right around May during the study. the year-long study period, and we see that at a few of the different sites, especially the hot spots. High concentrations of Enterococcus also coincided with high variation at each one of the stations, and this is why you see just like a temporary rise. Interestingly enough, Banyans, the site towards the middle, did exceed the 35 MPN per 100 milliliter state standard, but you didn't see the same pulse as you did at the other stations that were hotspots there. In conclusion, I would recommend prioritizing centralized wastewater infrastructure upgrades, and I'm not alone in this. The EPA just came out and did an assessment, and they have put in place a strict timeline for centralized wastewater infrastructure upgrades, and all around the island they've looked at the treatment plants, and they've established that urgent attention is needed in those places, and they're overseeing the reconstruction and the renovation of those plants. I want to thank the following people for all of their support throughout this project and their help. Thank you so much.
Very interesting. So Enterococcus is a very common, bioindicator, and I know it's very common on the mainland and more temperate environments. Do you think that are there other candidates, considering that we're in a tropical environment, for microbial bioindicators or species so instead of just looking at Enterococcus, are there other options, or is this really the best one for our environment?
Yeah, the broader study looked at nitrates and nitrites. They also looked at, I believe, staph as well in this study. And then I talked to somebody that had run a similar study on the Hilo side a few years ago, and he was actually looking for using eDNA to look at gut bacterium to trace that, because Enterococcus can come from a variety of different animals, birds, so to actually trace that to humans and human-sourced wastewater, it might be better to use gut bacterium.
Yeah, well. I'm just asking because it's really surprising that in the area that you saw that huge aggregation and density of cesspools, that was one of the lowest places for Enterococcus. So.
Right. I think that's because it might be coming from the centralized system. The line is running right there along the shore.
Did you all look for any underground mapping of water flow? That density could be there, but the water could be flowing left or right. It could be right below a high density, but not where it's outputting.
Yeah, that's interesting. I hadn't looked at that. I didn't get any of the data on like where the underground flow is going or which direction it might be flowing. So that's an interesting idea for sure. All right, thank you guys so much.